Hello everyone, my name is Gwanghoon Jang and I'm studying for a master's degree in Korean University. And today I'd like to talk to you about my research predicting mechanism of action of novel compounds using compound structure and transcriptomic signature co-embedding. First of all, as a bit of background, why you should care about mechanism of actions of compounds. Identifying mechanism of action of compounds is important in drug discovery process because it can improve on target efficacy and avoid potential side effects caused by off-target effects. And there are some approaches to identify MOAs of compounds. One of the most representative method is binding affinity assays. This method mainly focuses on the physical interaction between compounds and target proteins, so there is limitation that it neglects the impact of compounds on cellular phenotypes such as transcriptomic signatures. And another approach is using the transcriptomic signatures induced by compounds. A 1,000 dataset provides about 500,000 transcriptomic signatures corresponding to about 25,000 perturbations such as chemical compounds, gene knockdown, and gene knockout. And we can use this data to identify pathways affected by compounds. We can apply gene set enrichment analysis to infer pathway activity score. This is the overview of GSEA. GSEA needs length gene expression and gene set S, which consists of pathway genes. The score is calculated by working down the length gene expression, increasing a learning sum statistic when you encounter a gene in S, but decreasing it when you encounter genes not in S. The final enrichment score is the maximum deviation from zero encountered in the random work. But when you use transcriptomic signatures induced by compounds as an input of GSEA, there is a limitation that gene expression is the consequence of pathway activity, and an altered pathway is rarely presented in transcriptomic signatures of pathway genes. For example, in this figure, the true target of compound is signaling protein C, but in gene expression level, A, G, and H can be potential target. So to overcome this limitation, there's another approach is that using genetic perturbation signatures. Genetic perturbation signature is transcriptomic signatures of GP, such as gene knockdown and gene knockout. The approach is derived from the one hypothesis that if GP signatures are similar to a given compound signatures, the genes of GP signatures can be viewed as potential targets of the given compound. In the I-Link study, MOA pathways were identified based on the connectivity between compound signature and GP signatures. But there's one crucial limitation. All these methods fail to predict MOAs when the compound signatures are absent. This is motivation and challenge in our study. The goal is to identify MOAs of the novel compounds without transcriptomic signatures. And current limitation is that existing methods need transcriptomic signatures of compounds. So our solution is co-embedding model to represent compound structure and transcriptomic signature into the same latent space. Then embedding vectors of the co-embedding model can be used to replace transcriptomic signatures of compounds. This is the overview figure of our method, and first let's talk about co-embedding model. Our goal of Coembedding Model is to represent compound structure and transcriptomic signature into the same latent space. So the model uses two types of input data, chemical structure representation and transcriptomic signature. For the chemical structure representation of compounds, we used 2048-bit extended connectivity fingerprints, which represent topological fingerprints of chemical compounds. Each bit in ECCB indicates the presence of a chemical substructure. For the transcriptomic signature, we use a 1,000 dataset. A 1,000 dataset provides gene expression profiles at various normalization levels, level 1 to 5, And we use level 5 data that represent transcriptomic signatures for 978 landmark genes. The co-embedding model consists of two neural network encoder, structure encoder, FSTR, and signature encoder, FSIG. Structure encoder, FSTR, maps input compound structure XSTR with 2048 dimension to latent embedding vector ZSTR, which is 256 dimension. Signature encoder, FSIG, maps input signature XSIG, which is 978 dimension, to latent embedding vector ZSIG with 256 dimension. You can see the detail of the encoder model in this figure below. To encode compound structures and signatures simultaneously in co-embedding space, encoders are optimized with triple loss. While training with triple loss, anchor vector and positive vector are encoded close, and anchor vector and negative vectors are encoded far apart in latent space. So we set anchor vector as a compound signature embedding vector and positive vector as a compound structure embedding vector corresponding to the anchor compound, and negative vector as a compound structure embedding vector of any other compound. The optimization procedure allows embedding vectors to be located close to each other if their signatures are similar. Also, the compound structures and signatures can be mapped into the same embedding space. This co-embedding space enables the generation of embedding vectors that contain signature information, but only using its structure information alone. Next, MOA identification. First, we generate genetic perturbation embedding vectors. Signature encoder FSIG map inputs GP signatures to latent embedding vector. Then we calculate connectivity score. We define connectivity score as cosine similarity between a compound structure embedding and GP embedding vectors. Higher connectivity score indicate that two embedding vectors represent similar signatures, and for multiple GP signatures targeting the same gene, we only select the highest connected GP embedding for each gene. This is our connectivity scoring formula. For each compound, we rank GP genes based on their connectivity scores. Pathway enrichment analysis is performed with the ranked GP genes. We use a conventional GSE method for pathway enrichment analysis and KEGG 2019 as pathway gene set. After pathway enrichment analysis, we can get ranked MOA list with Pvalue and FDR score. Highly enriched pathways with statistical significance are considered as an MOA of a given compound. For the validation of co-embedding model, we assess whether connectivity associations between compound signatures and GP signatures can be reproduced in co-embedding space. We measure correlations between the connectivity scores of actual signatures and the connectivity scores of embedding vectors. Figure A illustrates the correlation distribution across 20,902 compounds, and Figure B illustrates regression plots for 4 compounds with highest correlation. We can observe that the strong correlation between actual signature and embeddings, and it shows that the connectivity association between compounds and GP can be retained in the embedding vector space. We evaluate whether the co-embedding model could generate embedding vectors that are informative for MOA identification. Compound structure embedding vectors of 20,902 compounds are visualized in 2D space. We can observe that compounds with similar MOAs are aligned close to each other. For example, compounds known as mTOR, PI3K, AKT inhibitors are clustered in the embedding space. And mTOR, AKT, and PI3K are topologically related to each other in signaling pathways. Overall, our analysis shows that the co-embedding model can generate compound embedding vectors useful for MOA identification. In order to quantitatively evaluate the performance of MOA prediction, we set baseline methods. First, TS978 use transcriptomic signature values of 978 landmark genes as length gene list of GSEA. TS12328 use all transcriptomic signature values of 12,328 genes. And TS connectivity use connectivity scores between GP signatures and compound signatures as length gene list of GSEA. And MOA, which is our method, use connectivity scores between GP embedding vectors and compound embedding vectors. For evaluation of MOA's robustness in various compound sets, we set the compounds into different sets. Evaluation set consists of all compounds whose transcriptomic signatures exist in 8,000, and unseen set consists of compounds excluded from training set of co-embedding model. And unseen hard set consists of compounds with the similarity less than 0.3 to training set of co-embedding model. And anti-cancer set consists of compounds with anatomical therapeutic chemical code of L01, which means anti-cancer. Novel set consists of compounds whose transcriptomic signature don't exist in 8,000, which means only MOA can predict MOA for novel compound set. True MOA pathway labels of compounds are obtained from KEGG drug database, and we evaluate the method based on p-value of true MOA pathway labels. This is p-value distribution of true MOA pathway in violin plot for each compound set. And this is the table of median p-value of true MOA pathways. We can observe that MOA achieves much lower median p-value than TS978 and TS12328. And it's comparable to TS connectivity for all compound sets, which means the MOA is able to accurately predict the MOAs of even further unseen compounds without using actual signatures. We further validate MOA with expanded MOA labels. First, we expand MOA labels by including pathways that contain target proteins of compounds. Target proteins of compounds are downloaded from clue.io. We compute area under receiver operating characteristic curve, AURC, for each compound. ROC curve is plotted by the true possible rate and against the false possible rate with various FDR thresholds. This is Boxplotter of AURC of expanded MOA labels for each compound set. This is median AURC of expanded MOA labels for each compound set. MOA significantly outperformed TS978 and TS12328. Moreover, MOA is able to achieve similar accuracy with TS connectivity despite not using actual signatures. We conduct a case study with a drug candidate named BI3406. Mutation in KRAS is known to be a major driver of some types of cancer, including non-small cell lung cancer, colorectal cancer, and pancreatic cancer. BI3406 is pan-KRAS inhibitor, and it binds to SOS1, which is an upstream protein of KRAS, and inhibit KRAS activity. We examine whether MOA can identify the last signaling pathway as a significant one with BI3406. Here's the result. MOA identifies last signaling pathway as a significant one with very low FDR, and pathway enrichment analysis with the actual signature fails to identify. Also, MOA can identify MAPK pathway and ERB pathway as significant ones. Interestingly, both pathway are downstream or upstream pathway of KRAS. In this study, we present MOA, a novel framework to identify MOAs or compounds without requiring actual signatures. And future research can improve mobile by utilizing more GP signatures. Increasing GP signatures can yield more robust pathway enrichment analysis. And mobile can be improved by identifying MOAs in a cell-specific manner. By training co-embedding model to learn compound structure embeddings in a cell-type specific way. And also, we expect mobile to be useful in generating mechanistic hypotheses for novel compound whose MOAs are unknown. Thank you for listening. Now we would like to move on to the live Q&A session.